International Neurotechnology Conference, which is held today and tomorrow in conjunction with the 3rd annual meeting of International Center for Neuroscience Research in partnership with the Georgian Neuroscience Association and Ivane Javakhishvili, Tbilisi State University in Georgia. And the main topic of the conference is a new technology application in mental and neurological disorders. Neurotech aims to bring together leading academic scientists and researchers to exchange and share their experiences and research results on all aspects of brain research technologies and neurotechnology. On behalf of the scientific committee, the scientists and actually MDs and something like this. So right now, because we're on the openings, I just want to ask Dr. Nasrollah Moradi-kor, the Director of International Center for Neuroscience Research and Institute for Intelligence Research from Tbilisi, Georgia, in order to come and give their speech.
Thank you, Helen. Hello everyone, I'm Nasrollah, Director of International Center for Neuroscience Research and Chair of the conference. Welcome all to International Neurotechnology Conference, Neurotech 2021. We are happy to see you again at this conference, my colleague and dear participant. The Neurotech Conference is organized by International Center for Neuroscience Research, Georgia, and supported by International Brain Research Organization, and Science Beam Company.
I'm going to ask Professor Dr. Indranath Chatterjee from South Korea, Department of Computer Engineering. Actually, right now, this is a keynote lecture. And right after the keynote lecture, we're going to have panel discussion, so you can ask your questions.
It's very easy and it's very comfortable for the psychiatrist or any kind of clinicians. So I cannot say that it can replace the doctor or the clinician, but it can actually help in the different kinds of medical intervention.
Right now, I'm just going to ask Dr. Mohamed Enamul Haq Chatterjee from the Department of Electrical Engineering from Qatar. Are you ready, Professor?
Then we wanted to think, okay, why EEG managed to separate the subject so well? Keystroke, okay, pattern is different. How EEG can do this? So we plotted the electrode signal coming from the brain area and how they are different from the subject to subject. So we identified that actually data coming from the brain of the subjects are from different part of the brain is contributing to this keyboard typing. This is a very well studied domain last 2 years. Maybe a few thousands of papers published in this domain. Even from our group, we published at least 10 plus papers on only X-ray CT domain to detect COVID.
Thank you.
Thank you.
I'm just going to ask Dr. Sawal Hamid Ali from the Department of Electrical, Electronics and System Engineering from Malaysia. Are you ready, Professor?
Yes, I'm ready. So this is the EMG signal acquisition. So this EMG signal acquisition shows the area for the contraction. And then the last one is the human emotion experiment. Okay, so basically in this human emotion experiment, okay, we are using in-lab experiment where we use audio visual as the stimuli. And then we also have done the simulator-based experiment where we use the driving simulator to identify the emotion. Okay, for this experiment, in this example, we just take data from 10 subjects. But for the other experiment, we are taking data from 64 subjects, right? And we can find a very specific pattern for each of the emotions.
Okay, thank you. Hello?
Yes, Professor.
Okay, thank you.
Right now, I'm just going to ask Professor Dr. Ahmed Yazbi in order to give their speech, the Professor of Pharmacology and Therapeutics from Egypt. Professor, are you ready?
Yes.
Thank you very much. So we looked at the retroperitoneal, an example of white visceral adipose tissue, and we looked at the epididymal adipose tissue as well. And again, we did not see an increase in either cytokine level. However, when we looked at the perivascular adipose tissue, which is the adipose tissue that surrounds the blood vessels and surrounds the heart, we found a huge increase.
I'm going to ask Professor Dr. Javakhishvili from Ivane Neurotechnology Center of Experimental Biomedicine, Tbilisi, Georgia, in order to give her speech.
So what was before and what was nowadays? I want to talk about the clinical part of EEG because it's very informative in neuroscience in clinical setting besides the psychology, because we use a QEEG, which is generates a map of activity and give us information about the abnormal states. And so it's very useful for patients when we, for instance, investigated patients during the pharmacotherapy.
Right now, I'm just going to ask Dr. Wael Mohamed from the Department of Basic Medical Science, Malaysia, in order to come and give his speech. Are you ready, Professor? Are you ready?
Yes, okay. So now could it be the ARC, which is the telepsychiatry? Okay, so now this is what we are proposing, or we are trying to propose as a solution now for Alzheimer's disease. It is a telepsychiatry, and from the name tele is telepsychiatry, psychiatry, which means using technology in psychiatry in general. So in general, telemedicine, it is intervention of a telecommunications device in the diagnosis and the overall care of patients that are separated from providers by distance. So
I'm just going to ask Dr. Syed Kumar from the Department of Physiology from Malaysia in order to give his speech. Are you ready, Doctor?
Yes, I'm ready. So this is elevated plasmase. So it's called elevated plasmase because it's elevated from the ground and it's in a plus shape. So the standard assessment for elevated plasmase is that we will be, in most of the studies, we'll be exposing the animal to this platform, and then we'll be looking into their behavior, which is based on the approach and avoidance concept, where the animals will be avoiding the open and the bright space from the open arm.
Right now, I'm just going to ask Dr. Sandra Diaz in order to give her speech from Germany. Are you ready, Doctor?
Based on rat or the mouse brain, which is part of the virtual brain, actually. So there is the virtual mouse brain as a part of the virtual brain, and you can get some connectivity data. In this case, the connectivity data was grabbed from the Allen Institute, and then there was a specific region in this mouse brain that was...
Right now, I'm just going to ask Dr. Arun Upadhyay, postdoctoral fellow, Department of Neurology, Northwestern University, Chicago, USA, in order to come and give his speech. Are you ready, Doctor?
Computer yes. Then we subjected ourselves for immunoprocess for those proteins individually. They're all different disease model proteins. So we saw that following the treatment of Nasrollah, these aggregates are cleared or degraded from the cells. And once we treat ourselves with chloroquine, which is an autophagy.
Okay, hello, Main.
Hi.
Hi. Good to meet you guys.
Thank you for your participation. We're waiting you.
When we focus on points with the highest amount of phase amplitude coupling modulation, we could see that the unattended case in all of these cases actually has a stronger, let's say, probability distribution function in terms of the difference between the peak and trough compared to the attended.
Theta, very sorry, but when we talk about the theta, we talk about the attention too, yeah? Generally. So generally, we teach students like this, yeah? Theta is responsible for attention, and when it's increased, it looks like some types of tension, yeah?
Yeah, that's true. So we have theta, and I mean, that I kept things very general in this talk because I didn't know how specific and
artificial intelligence using the they have been inspired from the neuroscience knowledge, and beside that, actually, the artificial intelligence also had influence on the neuroscience as well. So the artificial intelligence attempt is to make the computer perform more intellectual actions, and this type of the intellectuality has been used for the biomarker discovery from the clinical
using 18 days old embryos. Here you see some preparation. And after we were extracting brains and preparing neural culture. Papain was used for digestion of cortical cells